intriguing research area in lifespan extension. Critical analysis of existing material indicates a lot of problems and disappointingly little real successes in this field, which could at least partly be explained by absence of adequate coordination, in particular in development of modified models and criteria of assessment results, operative exchange of information and tools between interested research groups, etc. Let me very briefly present our vision of optimal strategy in such a research. It should be performed on 3-4 steps models, including screening experiments on short-lived species, preferentially Drosophila. This result should further refined and extended on rodent models, then maybe cell cultures, human cell cultures not shown in this table, and finally in humans. In this scheme, Drosophila is given the honor to be the first because of the well-known advantages, short lifespan included. In fact, it is the cheapest and quickest way to collect the initial information. It's understood that to carry out such widespread experiments is too difficult and expensive. For instance, I'm now in this kind of research for some 40 years, and never in my life I had a possibility to perform such an experiment. Still, we hope that ORION could be the one to start with. ORION is a multi-component complex drug based on derivatives of taurocolic acid and several other compounds. It was developed in the former USSR, allegedly for the party leaders and military involved in top secret missions. During the last years, it has been intensively studied in Ukraine in several academic institutions and clinics. In our Institute of Gerontology, we studied 2 modifications of ORION, ORION 10 and ORION 37. ORION 10 is designed to assess its effects on biological age of humans, and ORION 37 for experiments on animals. Unfortunately, our human sampling is too small to make final conclusions, but we have some exciting preliminary results. Some of them are summarized in this report prepared by the Clinic of Anti-Aging in Kiev, and who is interested, I can show after my presentation. In presentation, I will discuss only data of screening experiments on Drosophila. In this experiment, ORION was added into the feeding medium of lice in different concentrations, shown in percent, and studied from different ages. This first experiment was started on the 3rd day of imaginal life, and it's obvious that none of the tested concentrations actually improved lifespan. In experiments aimed to extend lifespan, there is the application of factors starting from early periods of life is the most frequently used scheme. However, from applied point of view, it has restricted significance because hardly anyone will risk experimenting on children. That's why we were more oriented on adult old animals in our research, like it is in this experiment started on the 41st day of life. Again, none of the studied concentrations of ORION really extended lifespan. However, when studied from 45th days of imaginal life, we observed some 30–50% of increase of mean remaining lifespan. It means mean lifespan when the starting age was accepted as 0. Moreover, when in experiments studied from 50 days of age, mean remaining lifespan increased 77%, and in experiments from 52 days, we had already 88% of increase. Finally, in this experiment, we registered highest increase of mean remaining lifespan, almost 3 times it was increased. And for this experiment from 61 days of age, there were more than doubling of mean remaining lifespan at 2 studied concentrations of ORION. Summarizing this part of result, it could be concluded that ORION could increase lifespan. However, there was a paradoxical age threshold. Only in experiments studied from second half of life, we actually registered the lifespan extension. There is another interesting moment in such a result. In this experiment, it concerns the longest living. In all our experiments, control animals, by the way, shown everywhere in a thick blue line for the sake of comprehension. Maximal lifespan of our control flies varied within 80-90 days. 80-90 days. None of experiments we registered for instance, flies live in 100 days. By the way, because 1 day of lifespan in Drosophila is roughly proportional to 1 year of human lifespan, such flies were named centenarians. And only when experiments were studied from 52 days, in experimental group we registered first centenarians. Some 10-20% of flies survived up to these ages, culminating super centenarians, real super centenarians in this experiment, when longest lived fly lived to 127 days. These experiments were so unexpected for us. I mean, how this subpopulation of super centenarians appeared, how it was induced, because they are absent in the control group, and could therefore be found ways to extend the proportion of these animals, so say not 10-20, but more percent of these animals. So those are intriguing questions we are trying to understand and cope with in our further research. But first of all, we would like to be sure that these phenomena are reproducible, and it's not experimental artifact, so we had to repeat and recheck these experiments. So the second series of experiments were performed. Unfortunately, in this series of experiments, some unexpected changes occurred in our flies. Control flies started living some 20 days longer. Even before those, they were long livers. 50-60 days of mean lifespan, it's enough for Drosophila, more than enough. But now our control flies live 80-90 days, in some experiments even 110-120 days. To my knowledge, this is the longest living flies are made. And it was additional challenge if ORION could extend lifespan in these already very long-living animals, because it's well known that in long-living populations it's extremely difficult to extend lifespan by additional interventions. So this is here are the results of our second experiment, studied from 50-50. If you remember, in the first experiment, 50-50 studied experiments gave almost 3 times longer mean lifespan, but not in this second experiment. You see, no life extending effect. It was the same when experiments were studied from 60 days. Again, no